So it's very different once the body transitions from a feasting or fed state to the fasting state, where we have no food ingestion. So when there's no food ingestion, then we start making use of our stores. So the glycogen that's been stored in the liver and muscle can actually be broken down into glucose, and then the glucose can be used to generate energy. So specifically, glucose is required to fuel the brain. So we can't generate energy in the brain from lipid. So glucose is the fuel that's specific to the brain. We can also use ketone bodies to fuel the brain, but here we're just focusing on the difference between fatty acids and glucose. So in the fasting state, we're going to start breaking down our fat to derive energy as well. So the fat will be broken down into the fatty acids and the glycerol, and those can be used to generate energy. So as opposed to above here in feasting, we're only generating the energy, well, the majority of the energy at least is coming from glucose, whereas in the fasting state, we're not only making energy from glucose, but also from fatty acids. So remember, in the first example of the fed state, the fatty acids were just being stored as fat. So now that we don't have as much glucose, we're actually relying on the fatty acids to derive energy as well. And then in the starvation state, we've used up all glycogen. So the glycogen doesn't actually exist anymore because we've used it all up. So there's no more carbohydrate available. So in this situation, the body is very reliant on the breakdown of fat to supply energy. So fat can be broken down into fatty acids, which can be directly used to make energy. But remember, fatty acid derived energy can't fuel some parts of the body. For example, the brain can't use the fatty acids to derive energy directly. So the fatty acids can actually be converted to ketone bodies, which can provide energy to the brain. So once we're in this starvation mode, we're not only using fat to derive energy, but also protein. So we're going to start breaking down our muscle proteins to get amino acids, which can then be converted into other fuel sources, for example, acetyl-CoA and glucose, which is from the carbon skeleton portion of our amino acids. So after we've taken off the amino group NH2 and removed that through the urea, the carbon skeleton can be used to derive energy. So essentially, in the feasting state, we're using the carbs to make ATP, and excess, whatever else there is considered excess, and that is basically going to be stored as fat. So this could be more carbs being stored as fat. The dietary fat and the protein will also lead to fat storage. Whereas in the fasting state, we use both the remaining carb and lipid, which has been previously stored, to generate our ATP. And in the starvation state, there's actually no carbs left, so we're reliant on the lipid and also protein. So we're going to be breaking down our muscle proteins now as well, so it's not very good, not a healthy state to be in. So we're relying on both of these to make energy. So you can see the usage of each macromolecule is different depending on whether we're in the feasting, fasting, or starvation state. So a few of the specific reactions that are going on. So looking here at panel B, the muscle glycogen can actually be broken down into glucose 6-phosphate. I know it says glucose here, but it actually is glucose 6-phosphate because in the muscle, we actually can't break it down into free glucose. And by doing this, so I see. So we've got glucose from the blood and also muscle glycogen contributing to the glucose, which can enter glycolysis and give us 2 ATP. So this is occurring in the muscle, and if the muscle is undergoing exercise, then it may be starved of oxygen. So that's where we get the pyruvate converted into lactate, which assists us in doing another cycle of glycolysis to get more energy, because we're making the NAD plus coenzyme that is required for the glycolysis process to keep going. So if there is oxygen present, then we can use that pyruvate that we created to generate more ATP in the common metabolic pathway, which is the citric acid cycle, electron transport chain, and oxidative phosphorylation, all of which occur in the mitochondria. But remember, this is oxygen dependent. So we can also have the fatty acids that are broken down feeding into the common metabolic pathway, and also the carbon skeletons of the breakdown from amino acids feeding into this common metabolic pathway as well. So we've talked a little bit about the role of glucose 6-phosphate already, but here we can see a plot of the carbohydrate processes that are either stimulated by insulin or glucagon. So those in red are stimulated by insulin, and those in blue are stimulated by glucagon. So insulin will promote the lowering of the blood glucose level. So to do this, it brings the glucose from the blood into the cell, which we can see here represented in yellow. Once the glucose is inside the cell, it can then be converted to glucose 6-phosphate, and from there we can convert it to pyruvate by continuing glycolysis, or we can actually store it as glycogen, which is what's being shown in this reaction. So we go first to glucose 1-phosphate, then UDP glucose, and then we're attaching an extra glucose unit to our glycogen chain. So we've got one extra unit on that chain. So all of these processes, the use of insulin either to make pyruvate through glycolysis or to make glycogen through glycogenesis, these are activated by insulin. Whereas if we want to actually break down our glycogen back into glucose, that's stimulated by glucagon. So we have the process of glycogen being broken down into glucose 1-phosphate and then going back towards glucose. We call this glycogenolysis. And once we have that glucose, it can actually be taken back out into the blood to raise the blood glucose level. So the gluconeogenesis is also controlled by hormones. So the gluconeogenesis is a glucose making process, which is going to be stimulated by glucagon. So ultimately, the gluconeogenesis is allowing us to convert pyruvate into glucose, and that glucose can then be taken out of the cell back out into the blood, and by doing this we're going to raise the blood glucose level. So why do we actually need to raise the blood glucose level? What would be the point in doing this? So we need to have enough glucose in the blood so it can be taken to organs and tissues that require glucose. So why would tissues and organs actually require glucose? They need it to make ATP to sustain their processes. Alright, so we can add in the extra element here of lipid metabolism on top of what we've already talked about with our glucose metabolism as a part of carbohydrate metabolism. So just like in carbohydrate metabolism, we have certain processes which are going to be activated by either glucagon or insulin. So I know we don't have glucagon listed for these processes, but glucagon will do the same as these hormones that I've circled here. So we've talked about it as glucagon and insulin, so let's just think of it as that. Okay, so let's firstly talk about what happens if we have insulin present. So if we have insulin present, that's promoting using glucose. But what does it do to our lipid? So it will actually promote making triacylglycerols, which can be stored as fat. So if we have the insulin present, then the acetyl-CoA from our glucose metabolism will actually be used to make fatty acids. So that's what we see in this reaction here. So the glucose is first converted to pyruvate using glycolysis, and then the pyruvate gets converted to the acetyl-CoA, and then that acetyl-CoA from carbohydrate metabolism can be used to make the fatty acids, which then combine with the glycerol to make triacylglycerate, which is our triacylglycerol. So that's how we can store fat, and that's promoted by insulin. Whereas the opposite to this process is basically breaking down lipids to be able to use them as an energy source. So the first part of this process is lipolysis, which is where we take our triacylglycerols and we convert those into the glycerol and also the fatty acids. So that's the arrows that we see here in green. So the triacylglycerol is being converted into fatty acids and also to glycerol. The glycerol can then be used in carbohydrate metabolism, and the fatty acids can actually be converted to acetyl-CoA through the beta oxidation process. So by creating these acetyl-CoA from our fatty acids, we're then able to feed them into the energy-making common metabolic pathway, starting with Krebs cycle, which connects to the electron transport chain, and also oxidative phosphorylation. And remember, a small amount of that acetyl-CoA from beta oxidation will also be used to make ketone bodies. So essentially, if we have glucagon, what is this going to do to our lipid? It's going to promote lipid to be converted into ATP. So we first break down the triacylglycerols to the fatty acids and the glycerol, and then we use those to make ATP through other reactions. Whereas the insulin, it will actually promote storage and / making of the triacylglycerols, so we can store those as fat. So that's where we have the acetyl-CoA from carbohydrate metabolism being used to make fatty acids, which can then combine with glycerol to be stored as fat. Alright, so here is our fed state. So we've already looked at quite a lot of these processes. We can see it all in one picture here. So here is what we're getting from digestion. So the digestion provides us with our glucose from our carbohydrates, the amino acids from the proteins, and the triacylglycerols from the fats and oils. So the amino acids can be taken to the liver to be processed and broken down, can also be used in the skeletal muscle to build up skeletal muscle. The glucose can be taken to different cells within the body that can derive energy from them, can also be taken to the liver. So once we have the glucose in the liver, it can be stored as glycogen, and if there's even more excess of the glucose, we can actually start converting it into the fatty acids to store it as fat, which is our triacylglycerol, which is taken to the adipose tissue. The triacylglycerides, or triacylglycerols as we've been calling them, will also be taken to the adipose tissue where we store them as fat. So basically we only really need the glucose here to derive some energy, although we don't see that very much in this explanation. But the others are going to be stored as fat. So we store the triacylglycerol as fat, we store the excess glucose as fat, and the amino acids that aren't being used to build muscle, they can actually be broken down, derive some energy from those, and that also links into fat storage. So our fasting state, there's a very different picture. This is where we're actually going to be breaking down all of our stores to derive energy. So let's start with our glycogen stores, which are in the liver and also in the muscle. So these are our first store that will be broken down. So we break down the glycogen into glucose, which can be used to make ATP. So we'll take the glucose to where it's required, for example, the brain or other organs, to derive energy from those locations. So once we've used up all the glycogen, then the amount of fat that we're breaking down is going to increase. So we'll start breaking down our triacylglycerol into the fatty acids and also glycerol, and via the blood they can be taken to where they're required to derive energy. So for example, the heart can derive energy from these fatty acids as well as from ketone bodies that we can make from the fatty acids via acetyl-CoA as well. So the muscle proteins will also be starting to break down if you're fasting for a very long period of time and entering starvation mode, and they can then be used to generate ATP by breaking down the amino acids as well into the keto acid form. So other tissues can also be a source of amino acids because other tissues are made up of proteins as well. So this is what we call tissue wastage when you're in a starvation state, where you start having to break down your tissues as well.